I'm going. I'm going, sir.
लग तो रही है साथ में खाएंगे ना
Hello, Dr. Chaturvedi.
Sir, good afternoon. Good afternoon, sir. How are you, sir?
I'm fine. Thank you.
Thank you, sir. Thank you.
How are you?
I'm good, sir. I'm doing good here. All your blessings.
Preparing for RSDI, eh?
Yes, sir. Yes, sir. You are coming, sir.
Hopefully.
Yeah, definitely, sir. We are waiting. And I think you will arrive on 21st, sir?
21st, yeah.
Yeah, 21st. Yes, sir. Shall we start, sir?
Puneet should be joining.
Yeah, I will remind him, sir.
He is the master teacher. I'm an extra man here. I'm ringing Puneet.
Good afternoon, sir.
Good afternoon, sir.
Namaste, sir. Dr. Sanjay.
Anyway, Mathur is here, so we can start now. Puneet will be
Namaskar.
joining very shortly.
Sir,
Yes, his phone has been discharged, sir, so he is joining from the second number. I have sent him the link again to his WhatsApp.
Okay. So good afternoon, friends. This is our second week of short certificate course, and I think we are going all right. Our Dean-elect Dr. Mathur is here, so I will ask him to say something about this program. Dr. Mathur, please.
thank you, sir. I think Dr. Jyotirmay Paul, President API, is not here in the program. Anyway, Dean Dr. Kamlesh Tiwari and friends, it's my pleasure to join today's program of short certificate courses. I wish to congratulate and compliment Dr. Kamlesh Tiwari, Dean Indian College of Physicians, for his welcome initiative and efforts to start a series of short certificate courses on the important subjects in medicine for postgraduate students and practicing physicians. I'm sure these courses will enrich and update the knowledge and skill of all the participants. Today's program is the second session of the short certificate course in diabetes mellitus, a very relevant subject of common clinical interest. The course director, Dr. Puneet Saxena, is an experienced teacher of high academic excellence and is a brilliant orator. I also would like to welcome today's speaker, Dr. Ashutosh, who is again a very young and brilliant academician with keen interest in teaching. I am sure the expertise of both Dr. Puneet Saxena and Dr. Ashutosh will add value to the program. My very sincere thanks to Dr. Kamlesh Tiwari for inviting me, and my best wishes for the success of the program. Thank you, sir.
Good afternoon, sir.
Sir, please unmute. Unmute, sir.
Yeah. Dr. Puneet Saxena
Yeah,
is
good
here.
afternoon.
First of all,
Yeah, good afternoon. Can you hear me?
yes, I can hear you.
Yeah, good afternoon, Tiwari sir. Good afternoon, Girish sir, and Dr. Ashutosh, and the whole team of Indian College of Physicians who is organizing this short certificate course. And I was just joined a few minutes late, and I was hearing Dr. Girish Mathur speaking. He is Dean-elect. And under the leadership of the API and ICP, these courses have been started. And we have seen that there's been an overwhelming response. And we are taking care of the diabetes short certificate course, and we have planned the curriculum in such a way that the all the basic topics of diabetes, right from the introduction to the complications and take home messages, are being covered. And I will request all the attendees to please pass on their names to the Dean ICP, so that your regular attendance can be marked, and later on when the exit exam is there, the exam can be conducted and your certificate can be issued well in time without any problems. So I will request all the attendees to attend all the seminar, all the classes of this short certificate course, and there will be roughly 20 to plus minus 2 classes as the course progresses on. So with that, I wish all the best to the all the attendees, and thank Dr. Tiwari to have given me this opportunity to start this course. Thank you very much.
Thank you, sir. Thank you. Now we can start. Dr. Ashutosh.
Yes, sir. So good afternoon all, and at the outset, I would like to pay my sincere thanks to Dean Sir, Dr. Kamlesh Tiwari Sir, President Dr. Jyotirmay Paul Sir, Dean-elect Dr. Girish Mathur Sir, my mentor, and Course Director Dr. Puneet Saxena Sir, Senior Professor and great academician, for giving me this opportunity. I also pay my regards to Joint Secretary, Dean's Place, Dr. Sanjay Kumar Sir, for being here, his valuable presence. And I welcome all the students, dear students, for this today's class. So I will just now start the screen sharing. I hope it is visible to all. Sir, is it visible?
Yes, sir.
Yeah.
Yes, it is visible.
Thank you, sir. Thank you. So I request all the attendees to kindly mute your audios till the lecture is going on. So welcome all, and today we will be discussing basics of the diabetes and glucose metabolism. So I am Dr. Ashutosh Chaturvedi, Associate Professor and Consultant Physician at Mahatma Gandhi Medical College and Hospital, Department of Medicine. Welcome you all. So dear friends, Today we will be talking about glucose metabolism and pathogenesis of diabetes. Now, first of all, in the first half, I will cover the basics of glucose metabolism that all of you must have read in your undergraduation in the biochemistry subject. Kindly mute your audios. I request again to all, I request the host to mute everyone. So what is metabolism? The entire spectrum of chemical reactions occurring in the living system. This is referred to as metabolism, and a metabolic pathway constitutes a series of enzymatic reactions to produce specific products. It can be divided into two categories, as we all know, that catabolism, that is the degradative process, and anabolism, that is the biosynthetic reactions. So catabolism is concerned with the breakdown of complex molecules to simpler ones with the release of energy, while anabolism is they comprise of biosynthetic reactions that involves the formation of complex molecules from the simple precursors. So it is the vice versa, opposite procedures. Now, today we will be dealing with the carbohydrate metabolism, and in that the glucose metabolism. So carbohydrates, as we all know, are the major source of energy for the living cells. Glucose is the central molecule in this carbohydrate metabolism, and all the major pathways of carbohydrate metabolism are connected with it. It is utilized as a source of energy. It is synthesized from non-carbohydrate precursors, and it is well stored as glycogen molecule to release as glucose whenever it is needed by our body. So the other monosaccharides which are important in carbohydrate metabolism are fructose, galactose, and mannose. So glucose, fructose, galactose, they are all monosaccharides. Now coming to the glucose molecule. So what do we want to know today? That we need to recognize the critical importance of glucose as the main carbohydrate of blood and main fuel of human cells. Please mute yourself. So recognizing the critical importance of glucose as the main carbohydrate of blood and main fuel of human cells. Secondly, recalling the sources of blood glucose in feeding and fasting states, recognizing the glucose transport as well into the cells, and also understand the basic concepts and directions of metabolism. So what is the general importance of carbohydrates in our body? What is the general importance of carbohydrates in human body? We need carbohydrates for energy, to provide energy through metabolic pathways, to store energy in the form of starch, to supply carbon for synthesis of other compounds that need carbon, and to form structural components in the cells and tissues. So this was the general importance. Now the critical importance of glucose, a constant source of glucose. It is an absolute requirement for human life. We all know this. It is the preferred energy for the brain. It is the required energy source for cells which have no or fewer mitochondria like red blood cells. And it is the essential source of energy for exercising muscles when we use them as a substrate for anaerobic glycolysis. So glucose as a preferred energy source, as a required energy source, and as an essential energy source, it is the most critical importance of this molecule. Now, metabolic pathways of glucose production and utilization, we need to say that like catabolic pathways and anabolic pathways. So some pathways, they produce the glucose, and in some pathways, the glucose is utilized. So glycogenolysis, gluconeogenesis, hexose interconversion, they are the pathways which produce the glucose, while in the HMP shunt, that is the pentose phosphate pathway, hexose interconversion and glycogenesis, the glucose is utilized. Also, there is glycolysis and Krebs cycle in which the glucose is metabolized. So this is the basic structure of glucose metabolism. Glucose, after glycolysis, it's converted into the pyruvate, or whenever there is the aerobic, it is converted into lactate. Then pyruvate goes to acetyl-CoA, NADH, FADH. And after electron transport change, flow of electrons, there is formation of ATP, which is the most essential requirement for energy of the body. So these are the basics. We are going back to the biochemistry subject in this session. And in the alternative pathway, the glucose by the HMP pathway, hexose monophosphate pathway, it converts to NADPH and ribose 5-phosphate, while after the glycogen synthesis, it is converted to glycogen. So this is the basic pathway. Now, catabolic pathways for providing energy, as I have discussed in the initial slide, that is glycolysis. Anaerobic glycolysis produces lactate, aerobic glycolysis produces pyruvate. And for providing synthetic products, we have the HMP pathway, that is the hexose monophosphate shunt, that produces NADPH and ribose 5-phosphate. So glycolysis, HMP shunt, these are the catabolic pathways, while in the synthetic pathways, we have the glycogen synthesis is there. So sources of glucose to human body, glucose can be obtained from 3 primary sources: carbohydrate in diet, by glycogen degradation, the process that is called as glycogenolysis. The glycogen which is synthesized from the glucose molecules, it is stored in liver and skeletal muscles. And in cases of fasting, the liver glycogen is degraded to yield glucose for the blood. In cases of muscular exercise, strenuous exercise, muscle glycogen is degraded to secure glucose for muscles as a source of energy. Similarly, in the gluconeogenesis, the glucose synthesis is there, and this is the synthesis of glucose from non-carbohydrate sources, like some amino acids are there, and it occurs in prolonged fasting. So in the initial stage of fasting, there is glycogenolysis. The glycogen which has been stored in the body, in the liver and skeletal muscles, the body utilizes the glycogen. And if the fasting is prolonged, then the gluconeogenesis takes place. That is the glucose synthesis from other non-carbohydrate sources like amino acids. So it is very simple. Now coming to sources of glucose from carbohydrate diet, we have free monosaccharides like glucose and fructose. Fructose is converted into glucose in liver. We have disaccharides like sucrose, lactose, and maltose. Glucose, sucrose is combination of glucose and fructose. Lactose has glucose and galactose, and maltose has glucose and glucose double molecules. So they are digested into monosaccharides in the intestine, and fructose and galactose are converted into glucose in the liver. Polysaccharides, we have starch, glycogen, they are digested into glucose in the GIT. So the summary, that is the major pathways of carbohydrate metabolism are glycolysis, the oxidation of glucose to pyruvate and lactate, citric acid cycle, the oxidation of acetyl coenzyme to carbon dioxide, gluconeogenesis from the non-carbohydrate precursors, glycogenesis, the formation of glycogen from glucose, glycogenolysis, that is the breakdown of glycogen to glucose, HMP shunt, which is an alternative pathway to glycolysis, and citric acid cycle for the oxidation of glucose. Then there is the uronic acid pathway, galactose metabolism, fructose metabolism, amino sugar, and mucopolysaccharide metabolism, which we are not discussing today. So we are mainly concerned with the glycolysis, gluconeogenesis, HMP shunt, glycogenolysis, and citric acid cycle. So let's discuss some of these in a very short, and just recall the our learnings from the undergraduate years. So glycolysis, this is also called as Embden-Meyerhof pathway, EM pathway, and it is defined as the sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. It takes place in all cells of the body. The enzymes of this pathway are present in cytosomal fraction, and it can be anaerobic or anaerobic. As we have already discussed, lactate is the end product of anaerobic glycolysis, while in aerobic conditions, pyruvate is formed, which is then oxidized to carbon dioxide and water. So what is the significance of this glycolysis? It is the major pathway for ATP synthesis in tissues that lacks mitochondria. It is the only source of energy in erythrocytes. That's a very important point. RBCs undergo glycolysis. In strenuous exercise, when muscle tissue lacks enough oxygen, anaerobic glycolysis forms the major form of energy for muscles. And it is also very essential for the brain, because the glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. Glycolysis is central metabolic pathway, and the intermediates of the glycolysis are useful for the synthesis of amino acids and fat. The reverse process of glycolysis will result in the synthesis of glucose, and that is called as gluconeogenesis. So the glycolytic pathway can be considered as the preliminary step before the complete oxidation takes place. Now comes the citric acid cycle. This is also known as Krebs cycle or the tricarboxylic acid cycle, TCA cycle. And this is most important metabolic pathway for the energy supply to the body. about 65 to 70% of the ATP is synthesized in Krebs cycle, and it involves oxidation of acetyl-CoA. This utilizes about 2/3 of total oxygen consumed by the body, and this is the most important central pathway that connects almost all individual metabolic pathways, either directly or indirectly, and supplies energy and provides many intermediates required for the synthesis of amino acids, glucose, heme, etc. Coming to HMP shunt, that is pentose phosphate pathway or phosphogluconate pathway. It is also an alternative pathway to glycolysis and TCA cycle for the oxidation of glucose. So the body has many channels. If the one channel has been stopped or if the process is not taking place, the body shifts to the other types of the mechanism for the metabolism, and it is concerned with the biosynthesis of NADPH and pentoses. The enzymes of HMP shunt are located in the cytosol, the tissues such as liver, adipose tissue, adrenal gland, RBCs, testes, lactating mammary gland. They are highly active in HMP shunt. The HMP shunt takes place very frequently in these organs. So most of these tissues are involved in the biosynthesis of fatty acids and steroids, which are dependent on the supply of NADPH. So you can understand that how the catecholamines, steroids, they are dependent on this HMP shunt pathway. Their production occurs through this HMP shunt. So it's a very important pathway also. Now, the gluconeogenesis, the synthesis of glucose, as the term indicates, gluconeogenesis. So synthesis of genesis of glucose from neo means new. So new means non-carbohydrate compounds. That is called as gluconeogenesis. And the major substrates are lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. Gluconeogenesis occurs mainly in the cytosol, and although the precursors are also produced in the mitochondria and mostly takes place in liver and kidney matrix. So what is the significance of this gluconeogenesis? Brain and CNS, RBCs, testes, and kidney, the medulla of the kidney are dependent on glucose for continuous supply of energy. So human brain alone requires about 120 gram of glucose per day out of this 160 gram needed by the entire body. So you can imagine how important is the glucose for brain. Out of 160 gram, 120 gram is alone required by the human brain, and glucose is the only source that supplies energy to the skeletal muscle also under anaerobic conditions. So in fasting, even more than a day, gluconeogenesis must occur to meet the basal requirements of the body for glucose and to maintain the intermediate of the citric acid cycle. And this is essential for the survival of humans and other animals in the prolonged fasting stage. So certain metabolites produced in the tissues, they accumulate in the blood, that like lactate, glycerol, propionate, and gluconeogenesis clears them from the blood. So this is the importance of gluconeogenesis. Now, after the glucose metabolism, I will shortly describe the organs involved in the glucose homeostasis. What is the importance of these organs? So 4 organs chiefly are involved in glucose metabolism: liver, muscle, adipose tissue, and kidney. So liver does not need insulin to facilitate glucose uptake. However, insulin plays a key role in the regulation of glucose output by the liver. It prevents gluconeogenesis and breakdown of stored glycogen. And in the fed state, liver removes about 70% of the glucose load that is delivered by portal vein from the gut. And that is partly oxidized and partly converted to glycogen for next use as a fuel under fasting conditions. Glucose exceeding these requirements is metabolized by the liver into fatty acids and triacylglycerols, which are incorporated into VLDL and transported into adipose tissue stores. So this was in the fed state. Now in the fasting state, blood glucose concentration is maintained by liver glycogen breakdown, that is glycogenolysis, while glycogen stores last for several hours, and then by gluconeogenesis, that is from glycerol, lactate, pyruvate, and gluconeogenic amino acids, as we discussed just now. So after liver, the muscle is responsible for majority of glucose uptake, that is 75 to 80%. And once glucose enters muscle cells, it can be used immediately for energy or stored as glycogen, whatever is the requirement of the body. So insulin activates this glycogen synthase enzyme, that is which chiefly regulates the production of glycogen. So muscle, then comes the adipose tissue, which is the major reservoir for excess glucose storage in the form of triacylglycerol, TAG. Insulin promotes the synthesis of TAG by activation of lipoprotein lipase enzyme in the capillary walls of adipose tissue and by inhibiting hormone-sensitive lipase within adipocytes and thus preventing the hydrolysis of this stored molecule. So during the fasting state, muscle and adipose tissue adapt to the oxidation of fatty acids for energy purposes. Now, you must be surprised if you want to recollect that kidney has also a role in the glucose homeostasis, and it is involved by three mechanisms. First is the release of glucose into circulation via gluconeogenesis. Secondly, it also facilitates the uptake of glucose from circulation to satisfy its own energy needs. And finally, the reabsorption of glucose in proximal tubules where SGLT1 and SGLT2 receptors and GLUT1 and GLUT2 receptors are located. So this is the very important point in the management of diabetes these days. We all know that SGLT2 receptor drugs are now the main armamentarium, main weapon in the management of diabetes. So more glucose may be excreted in the urine if the concentration in filtered urine exceeds the threshold of glucose reabsorption. So this is the importance of kidney in the glucose homeostasis. Now, hormones regulating glucose homeostasis. The regulation of glucose metabolism is under the influence of several hormones whose target tissues, as we all know, are liver, muscles, and adipose tissue. And only insulin lowers the postprandial blood glucose concentration by facilitating glucose entry into the cells. In addition, it also supports the synthesis of many substances that leads to its anabolic effects, that is weight gain, anabolic effects. The effect of glucagon, growth hormone, cortisol, and adrenaline on blood glucose is the opposite. These are the counter-regulating hormones, and they increase their concentration in blood at low glucose concentrations and provide sufficient energy substrates from glycogenolysis, gluconeogenesis, lipolysis, and ketogenesis. So whenever there is hypoglycemia, these hormones like glucagon, GH, cortisol, they all get activated and their concentration increases in the blood. And then this substrate, they provide energy substrates, alternate of glucose from these processes, as we have initially discussed, like glycogenolysis, gluconeogenesis occurs in brain, lipolysis, ketogenesis. So these supply glucose at the critical times. So this is the summary of insulin, glucagon, adrenaline, cortisol, growth hormone, the processes of the liver like glycogenolysis, gluconeogenesis, ketogenesis, muscles. So this summary of the effects of various hormones on glucose metabolism, and we will be discussing the insulin metabolism and about the insulin and glucagon hormone in our subsequent slides. So now coming to how does the glucose metabolism interferes with the genesis of the diabetes, that is, how does diabetes disrupt the homeostatic level of glucose in the blood. So for this, we have to go into the anatomy and physiology of the pancreas that we will discuss in the subsequent slides. So the pancreas is approximately 20 centimeters in length and it weighs about 80 grams. The pancreas has two major types of tissue. This is very important to notice that it has two major types of tissues. First is the acini that secrete the digestive juices into the duodenum. And second is the islets of Langerhans that secrete insulin, glucagon, and other hormones directly into the blood. So human pancreas has about 1 to 2 million of islets of 0.3 mm in diameter, although the islet account for only about 1% of pancreatic cell population. So this is the anatomy of the pancreas. As you can see, there is the head, neck, body, and tail, 4 parts. And fifth one is the uncinate process that combines at the duodenum. From this uncinate process, the pancreas get joined to the duodenum, and these are the pancreatic ducts. Gallbladder, which is attached here. This is the common bile duct, accessory pancreatic duct, the opening of accessory pancreatic duct, and duodenal papilla in the duodenum. So if we go into the detailed histology and microscopy of these pancreatic tissue, so here you can see the acini are distributed. These are the ducts, and in the ducts we have two types of tissue. This is the acini. It's all around. This is acini, and the central structure, this is the islet of Langerhans. So if we enlarge this islet of Langerhans, we get this, and the islet of Langerhans is comprised of beta cell, delta cell, alpha cell, and pancreatic polypeptide, that is the PP cell. So alpha cells of these islets of Langerhans, they secrete glucagon, which elevates blood glucose concentrations. Beta cells secrete insulin that reduce glucose concentration. We all know delta cells secrete somatostatin, which slows the rate of food absorption and digestive enzyme secretion, while as the F cells and TP cells, they secrete the pancreatic polypeptide. So there is close interrelation among various cell types that allow regulation of secretion of some hormones by other hormones. So the islets of Langerhans, as we discussed, it contains of 4 types of hormone secreting cells. Alpha cells comprise of 25% of composition, beta cells are 60%, delta cells 10%, and PP cells 5%. So these islets are organized around small capillaries into which the cells secrete their hormones. So this was the pancreatic structure. Now, how do the liver and pancreas allow us to utilize the nutrients that we consume? So the glucose is the preferred nutrient and carbohydrates are the preferred source of energy. Final products of carbohydrate digestion are monosaccharides like fructose, and galactose. And glucose is a large molecule that must be broken down into a form of energy that is utilized by the cell as ATP. So much of fructose and all galactose are converted to glucose in the liver and released back into the blood. So these are the pancreatic islet peptide products. Insulin that functions, the main functions of insulin are that it decreases the gluconeogenesis, glycogenolysis, fatty acid breakdown, and ketogenesis. It increases the glycogenesis, the conversion of glucose into glycogen, and it increases protein synthesis, and that's why it leads to weight gain. It has an anabolic function, and it also increases the glucose uptake. Glucagon has opposite effects of insulin, and this is secreted by alpha cells. It increases hepatic glycogenolysis and gluconeogenesis. Somatostatin secreted by delta cells. It inhibits GI secretion, inhibits secretion and action of all GI endocrine peptides, and inhibits the cell growth. While pancreatic polypeptides, it inhibits pancreatic exocrine secretion and secretion of insulin and facilitates hepatic effects of insulin. Other enzymes, other hormones like amylin, pancreastatin, and ghrelin are also there. So coming to metabolic efficiency of glucose, the cells rely on insulin for efficient absorption of glucose from the blood, except brain cells. Insulin also enhances ATP production. Insulin is a small protein composed of 2 amino acid chains connected by 2 disulfide linkages. It is secreted by beta cells and it circulates in blood in unbound form, and it has a plasma half-life of about 6 minutes and is cleared from circulation in 10 to 15 minutes. It doesn't combine with receptors, and in target cell, it is degraded by insulinase, mostly in liver and sometimes in kidneys and muscles also. In target cells, it is also degraded by connecting peptide and jet joins the 2 chains. So it is a protein of 21 amino acid A chain and 30 amino acid B chain that is linked with the disulfide linkages. So the mechanism of action of insulin, the receptor is a combination of 4 subunits held together by disulfide linkages, 2 alpha subunits lying outside and 2 beta subunits protruding into the cell cytoplasm. When insulin binds to the alpha subunit in the targeted tissues, the beta subunits in turn become activated. And this activation of beta subunits triggers a series of reactions that draw the glucose transport enzyme into the cell membrane. Now cells like cells of liver, muscle, adipose tissue, but not brain, they are now able to increase their uptake of glucose after this activation of beta subunits takes place. So within seconds after insulin binds with this membrane, the cells are able to increase the uptake of glucose and the reactions are facilitated. So the cell membrane also becomes more permeable to many amino acids after this beta subunits activation takes place. Now only enough ATP for immediate cellular requirements is made at any one time. So glucose that is not needed, the leftover glucose which is not utilized for ATP production is now anabolized into glycogen by insulin, and it is stored for later use in liver and muscle. So it is how the insulin facilitates the anabolic action that converts glucose into the glycogen. Also, glycogenesis, the synthesis of glycogen from glucose molecules. Insulin also stimulates glycogenesis and inhibits glycogenolysis. So why glucose is stored as glycogen? Because glucose is in liquid form. As the number of glucose molecules increases, the pressure inside the cell increases, and that converts glucose to glycogen in solid form that relieves the pressure inside the cell. Glucose conversion also takes place into the fat. That is excess glucose that is preferentially stored as glycogen, but when cells are saturated with glycogen, that is, liver cells store 5 to 8% of their weight as glycogen, while muscle cells only about 1 to 3%. Then the remaining additional glucose is converted to fat in the liver and is stored as fat in the adipose cells. So this is the fascinating mechanism and metabolism of insulin and glucose interconnected. Now, insulin also promotes the conversion of all glucose excess in liver that cannot be stored as glycogen into fatty acids also. Insulin also has a role in protein metabolism. That is, insulin, like growth hormone, stimulates transport of amino acids into the cell. And it also increases the translation of messenger RNA, that is mRNA, and thus forming the new proteins. Now, insulin and growth hormone, it interacts synergistically to promote growth. It also stimulates the absorption of fatty acids and glycerol by adipocytes, where they are stored as triglycerides. So to summarize the metabolic effects of insulin, it increases the rate of glucose transport into target cell, increases the rate of utilization, glucose utilization and ATP formation, increases the conversion of glucose to glycogen in liver and skeletal muscle, increases amino acid absorption, protein synthesis, also increases the triglyceride synthesis in adipose tissue. It decreases the high blood glucose levels. Insulin is an anabolic hormone. So it's a very much more clear slide to represent the insulin effects. There are left ones are the negatives, that is stops gluconeogenesis, glycogenolysis, lipolysis, ketogenesis, proteolysis, while it facilitates glucose uptake in muscle and adipose tissue, glycolysis, glycogen synthesis, protein synthesis, and uptake of ions, especially potassium and phosphate ions. So after insulin, the second most important hormone in the glucose metabolism is the glucagon. That is the most potent regulator of glucagon secretion, is the circulating glucose itself. Hypoglycemia stimulates the pancreatic alpha cells to release glucagon, and hyperglycemia inhibits the glucagon synthesis. So it is vice versa. Glucagon promotes hepatic conversion of glycogen to glucose by glycogenolysis. It stimulates de novo glucose synthesis by gluconeogenesis, and it inhibits glucose breakdown by glycolysis and glycogen formation, that is glycogenesis. So very important hormone after insulin is the glucagon, which has many functions. A few words about somatostatin, that is a polypeptide with 14 amino acids with a 3-minute half-life, and it's secreted by delta cells. It depresses secretion of insulin and glucagon both. So this is the important point. Somatostatin reduces the secretion of insulin and glucagon. It also reduces the motility of stomach, duodenum, and gallbladder, and decreases secretion and absorption in the GIT. It extends the period of time for assimilation of food. So this is important. Pancreatic polypeptide are secreted by F cells. They are concerned with the gallbladder contractions. So they inhibit the gallbladder contractions and regulates the production of some pancreatic enzymes, and may also help in the controlling the rate of nutrient absorption by the digestive tract. So this was the first part, dear friends, that we discussed quite exhaustively the glucose metabolism and some about the hormones, mechanisms of the glucose metabolism, and how it is concerned with the genesis of diabetes mellitus, the production, the anatomy, physiology of pancreas and its hormones, insulin, glucagon, etc. Now coming to the second part of this lecture, that is the pathogenesis of diabetes mellitus. But I must compliment Dr. Tiwari sir, Dr. Puneet Saxena sir, to design this lecture that simultaneously with glucose metabolism, and now we are discussing the pathogenesis of diabetes. This is very important to know the basics of glucose metabolism. So dear friends, now coming to the clinical part of this lecture, diabetes mellitus, it's a disorder of carbohydrate metabolism, which is characterized by impaired ability of the body to produce or respond to insulin and thereby maintain proper levels of glucose in the blood. So this is the basic definition of diabetes. The term diabetes mellitus, it refers to the excretion of large quantities of sweet urine, as described by Banting and Bess, which are pioneers which have been the pioneers in the field of diabetes. Diabetes is an old Greek word that is taken from diabano for siphon and means diaeresis. Mellitus, that has been taken from Latin word that means mel, means honey or sweet taste of a urine. So diabetes mellitus means the diuresis or of the sweet taste of the urine. Now coming to types of diabetes mellitus, type 1, type 2, gestational, and other many types of diabetes. Like, please mute yourself. Please mute yourself. Coming to the types of diabetes, that is type 1, type 2, gestational, and other types of diabetes like MODY and LADA. And these types of diabetes mellitus, this classification will be taken up in the subsequent lectures by other renowned faculty. So pathogenesis of type 1 diabetes, it is an autoimmune disease in which islet destruction is caused primarily by immune effector cells that reacting against the endogenous beta cell antigens. So that occurs in 10% of the cases. And for a broader classification, type 1 diabetes has been further subdivided into subtype 1A and subtype 1B. Subtype 1A is due to autoimmune destruction of beta cells, while subtype 1B is idiopathic, that we doesn't know about its cause. So what are the main factors responsible for the genesis of diabetes mellitus type 1? First and foremost is the genetic susceptibility. Second is the autoimmunity. And third is the environmental factors. There are a lot of environmental factors. We will discuss them. So what are genetic factors? Half of the cases have genetic predisposition to type 1A DN. And they have a gene in HLA, that is human leukocyte antigen region of chromosome 6. That is MHC class 2 region. So it is important that chromosome 6 is involved in the genesis of type 1 diabetes, and the sites are HLA-DR3, HLA-DR4, or HLA-DQ locus of the chromosome 6. So this is the HLA complex. This is the chromosome 6, long arm and short arm. In the short arm, there's located the HLA region, and these are the regions involved in this HLA region. We have the DP, DQ, DR, B, C, and A. So these are the class 1 regions, A, B, C, and these are the class 2 regions, DP, DQ, DR. So DQ, DR are involved in this, and apart from this, DQ and DR3 and DR4 are involved. Other genetic factors that are responsible are the insulin gene polymorphisms, which occur on the short arm of chromosome 11. They also account for about 10% of the cases. And the cytotoxic T lymphocyte associated antigen 4 gene, that is CTLA4, that is placed on the long arm of chromosome 2. So the chromosome 6, chromosome 11, and chromosome 2, they harbor the genetic factors, they harbor the polymorphisms of the insulin gene that produces the type 1 diabetes. Now, secondly, autoimmunity. The second factor is the autoimmunity, that is the presence of islet cell antibodies against glutamic acid decarboxylase, GAD, that we routinely test in the type 1 diabetes patients in our clinical practice, or insulin present in the beta cells. So the occurrence of lymphocytic infiltrate in and around the pancreatic islets, which is termed as insulitis, it chiefly consists of CD8 cells, and with variable number of CD4 plus T lymphocytes and macrophages also. So the another theory is the selective destruction of beta cells, while other islet cells types like alpha and delta cell remain unaffected. This is mediated by T cells. Association of type 1A diabetes with other autoimmune diseases has also been recognized, and commonly we encounter in our clinical practice, like it is also associated with Graves' disease, Addison's disease, and Hashimoto's thyroiditis. So as you can see here, these are the normal islets versus islets that has insulitis. So these are the normal islet tissue, secretory acini, connective tissue capsule, capillaries, and these are the pancreatic islet cells. Now, if you can see here, these invading white beta cells, they are invading here the insulin producing beta cells, and this the normal architectural pattern has been distorted in this insulitis patient. So after the genetic and autoimmunity factors, we will now come to environmental factors. The environmental factors involved include like viruses like rubella, coxsackie virus, enteroviruses. They have been recognized as a causative agents of type 1 diabetes. And recently we have the COVID outbreak. So what was that? That was also the environmental factors. A lot of patients, we have seen that after they have suffered from COVID, although they have not been treated with the steroids, but they have developed the diabetes has been developed in many COVID patients. So these are the environmental factors that leads to, although this is type 1, but the type 2 factors in type 2 diabetes, environmental factors are also there. And the second possibility is the relationship of early exposure to bovine milk proteins and also occurrence of autoimmune process in type 1A diabetes. Experimental induction of type 1 diabetes with certain chemicals has been possible, like chemicals like alloxan, etc. So these environmental factors are also important. Now, this is the autoimmune basis for type 1 diabetes. There is beta cell mass. Now, immune dysregulation is started. There is interaction between genes imparting susceptibility and resistance. Environmental triggers and regulators also go on there, and that leads to variable insulinitis and beta cell sensitivity to injury. And that phase is called as pre-diabetes. So after the loss of first phase insulin response, that is the glucose tolerance, there develops the glucose intolerance, and now this pre-diabetes is further then converted into overt type 1 diabetes, and where the C-peptide goes undetectable. So this is the autoimmune basis for type 1 diabetes. Various models have been suggested for the pathogenesis of type 1 diabetes. Here you can see that this is called as primary chronic progressive pattern. Here beta cell mass is initially it is beta cell mass is there, but then this simultaneously chronically progresses, and over the time beta cell mass is gone. And secondly, this is the benign model that beta cell mass fluctuates up and down, up and down. Third one is the relapsing remitting model, and fourth is the secondary chronic progressive model. Over the time in everybody, the beta cell mass goes down and it is finished, except few cases of this benign model where the beta cell mass is quite maintained after a zigzag pattern. So how type 1 diabetes might arise? So there you can see genetic susceptibility is the first and foremost starting point. HLA, insulin, and other pathways. Then the environmental trigger is there by various bacteria, nutrition, or chemicals. Then the initial CD8 kills this solidification by APCs, and then beta cell autoantibodies, CD4, CD8 responses come into account, and then epitope spreading, and then finally it is the clinical diabetes where you can find the autoantibodies, insulin, GAD, ZNT8, CD4 cells, CD8 cells become activated, beta cell proliferation is there, and the final leads to the clinical diabetes. So it's a very, very long and interesting process, taking about years to develop. So these are the points where we can understand that in why the type 1 diabetes is common in a wide range of the age years. Like it starts from neonatal diabetes, and we can we find it in many patients up to the age of 25 or 30 years, because this pathway takes time, and it is individual. It is also individualized. Like treatment, the pathogenesis is also individualized. How somebody's body may respond to this genetic susceptibility and the environmental triggers. God knows this, and we can only help our patients by treatment. So to summarize, the genetic susceptibility and after environmental factors like viral infections, experimental induction with chemicals, bovine milk proteins role has been suggested, geographic and seasonal variations, then the autoimmune factors, islet cell antibodies, insulitis, and then leads to the selective destruction of beta cells, and it finally generates the type 1 diabetes. So this is the pathogenesis of type 1 diabetes. Now coming to the pathogenesis of type 2 diabetes, the development of type 2 diabetes, it also involves two key abnormalities, that is insulin resistance and beta cell dysfunction. Now, insulin resistance is the decreased response of peripheral tissues, especially skeletal muscle, adipose tissue, and liver to insulin. While beta cell dysfunction, this is the inadequate insulin secretion in the face of insulin resistance and hyperglycemia. So, in most of the patients, both go simultaneously. Insulin resistance is also there. Beta cell dysfunction is also there. So, this is the insulin synthesis and secretion, and most important stimulus for insulin synthesis is the release of glucose itself. It is uptake by a glucotransport, that is insulin-dependent glucose transporter. These are the cells, and this is the pathway. Action of insulin, we have discussed in detail that in the muscles, the glucose uptake increases, glycogen synthesis increases, protein synthesis increases, while in the liver, the insulin facilitates the glycogen synthesis and lipogenesis, while it inhibits the gluconeogenesis. In adipose tissue, insulin increases the glucose uptake, increases lipogenesis, and decreases lipolysis. So, how the incretins, what are incretins and how they are involved in this pathophysiology of diabetes? They are the gut-derived peptide hormones that are rapidly secreted in response to a meal that in turn induces insulin secretion. They act on G protein coupled receptors which are present on beta cells. And the two most important incretins are GIP and GLP-1, which we all know that glucose-dependent insulinotropic polypeptide and glucagon-like polypeptide GLP-1. So their function is to increase insulin secretion from beta cells and reduce glucagon secretion from alpha cells. And they also delay the gastric emptying. While in the degradation, its degradation happens in circulation via DPPs. We all know dipeptidyl peptidases, and incretin effect is blunted. This incretin effect is blunted in type 2 diabetes. So the DPP4 inhibitors that we commonly use in our practice, which are also called as gliptins, they are the approved drugs to treat type 2 diabetes, and they act via this incretin mechanism. So the main factors responsible for pathogenesis of type 2, genetic factor, constitutional factor, and in the genetic and constitutional factors, they both combine to produce insulin resistance and impaired insulin secretion. So the most common, there are two pathways, insulin resistance and impaired insulin secretion. Although genetic and constitutional factors are also taking part in the pathogenesis of type 2 diabetes. And we should know that as a person with one parent having type 2 diabetes is at an increased risk of getting diabetes, while if both parents are having type 2 diabetes, the risk in the offsprings of diabetes mellitus, it rises to 40%. Certain environmental factors such as obesity, hypertension, and level of physical activity, they also play a role in the expression of these diseases. And this is very important in today's time where we are all, the majority of population is having the sedentary lifestyle. So insulin resistance, it is the resistance to action of insulin that impairs glucose utilization, and hence hyperglycemia is there. There is increased hepatic synthesis of glucose, and the clinical features consist of hyperglycemia, compensatory hyperinsulinemia, cardiovascular disease not occur at this stage. There's mild hypertension, dyslipidemia, which we call as diabetic dyslipidemia, may be there, like increased LDL, reduced HDL. So insulin resistance leads to obesity and also affects peripheral tissue sensitivity towards insulin, and it is also an important pathway. Now, after insulin resistance, the second path is the impaired insulin secretion, because hyperglycemia and compensatory hyperinsulinemia are there, and eventually there is failure of beta cell function to secrete adequate insulin in cases of type 2 diabetes which have mild to moderate deficiency. The islet amyloid, that is the amylin we had discussed shortly in our previous slides. That is in long-standing cases of type 2 diabetes, the amylin may be responsible for impaired function of beta cells. And also the chronic hyperglycemia that surrounds the islets leads to glucose toxicity, and it may paradoxically impair the islet cell function. So, dear friends, you can see the amyloidosis of pancreatic islet tissue. The islets are mostly replaced by structureless eosinophilic material, which stains positively with Congo red. So this is the normal tissue, and here you can see the destroyed islet cells, which have been replaced by eosinophilic material. So to summarize the type 2 diabetes pathogenesis, first of all, genetic factors, which are already there in the newborn or in the person. The concordance in identical twins is 80%, while both parents diabetic, there is 40 to 50% risk to the offspring. Constitutional factors, obesity, hypertension, sedentary lifestyle, low physical activity, they all lead to insulin resistance. Genetic factors also play an important role in insulin resistance. And genetic factors also leads to decreased insulin secretion. So in the decreased insulin secretion, amylin, glucose toxicity of islets, and lipotoxicity, their role have been discussed and postulated in this pathogenesis. And from the insulin resistance, there are receptor and post-receptor defects that impair glucose utilization. So both these combine and they lead to increased hepatic glucose synthesis and leading to subsequent hyperglycemia and development of type 2 diabetes mellitus. So this was the summary of pathogenesis of type 2 diabetes. So let's discuss very, very quickly the pathogenetic and clinical difference of type 1 and type 2D. And this is very important. And we can finally summarize the lecture here that you can see that in type 1, the age that is the lowest, they written in the lowest part, age, young, that is mostly under 35. Type 2 diabetes is old and middle age is there. Now, beginning of disease acute in type 1, while there is gradual onset in type 2 diabetes. Duration is very, very labile in type 1, while duration is very stable in type 2. Ketosis, ketoacidosis, they often develop, more common in type 1, and it is rarely found in type 2, except whenever there is the severe infection, sepsis, etc. are there. So the body weight in type 1 decreases or it remains normal, while you can find obesity in 80 to 90% of type 2 diabetes patients. The treatment is insulin diet in type 1, control of diet, and type 2, diet, drugs along with the insulin if the drugs are unable to achieve the expected HbA1c goal. Degree of severity, type 1 diabetes moderate to severe, while in type 2 the patients may be mild, moderate or severe hyperglycemia. Now, connection with HLA system, we have seen in HLA DR, DQ region, it is there in type 1, it is absent in type 2. Level of insulin C-peptide is decreased or absent, almost absent in type 1, where it is frequently normal in the type 2 diabetes. About the antibodies to beta cells, they are present in 80 to 90% of patients on first week or month in type 1, while they are absent in the type 2 diabetes patients. Late complications, microangiopathies may rise in type 1, while there are macroangiopathies more commonly seen in type 2. Mortality less than 10% and more than 20% according to various studies. 10 to 20% spreads, whereas in 80 to 90% in type 2. So the pathogenesis concept to finally insulin resistance occurs early before glucose intolerance. Genetic causes are there, obesity, aging, lifestyle. If have a healthy beta cells are there, then they can compensate and remain euglycemic. But if the beta cells are susceptible, then beta cell dysfunction results in imperfect compensation and they progress to pre-diabetes stage. And there is onset of acquired abnormalities also. So this hyperglycemia further worsens and leads to type 1 or type 2 diabetes. And then this is a vicious cycle which is followed in the body and finally terminating in the complications of diabetes mellitus. So dear friends, dear delegates, dear faculty, my mentors, thank you all for being there this afternoon and being patient. I thank you, Dean Sir, Dean-elect Sir, President Sir, and Course Director Dr. Puneet Saxena Sir for giving me this wonderful opportunity to deliver this honor lecture here. Thank you, sir.
Thank you, Astos. Thank you very much. Now I will like Dr. Sanjay. Dr. Sanjay, Joint Secretary. Is he here? Yes. Your comment, please, Dr. Sanjay. You are muted. You are muted.
Really, it was a wonderful lecture, right from the basics of the glucose metabolism to the pathogenesis of different type of diabetes and how, because the learning of these points are very much important in order to understand the treatment profile of the different types of diabetes and how we can manage the diabetes in a better way. So it was really a wonderful lecture. Not only the PG students learned, I also learned a lot of points. Thank you.
Any question from students? Any question? You can write on chat box. No? Thank you, Astos. Thank you very much.
Thank you so much, sir. Thank you so much.
Thank you.
Thank you, Dr. Ashutosh.
Thank you, Sanjay sir. Thank you very much for your presence. Thank you.
So we have